All right. Hello, everyone. A very warm good evening to all of you. Thank you so much that you took time to tune into our webinar today. And like always, like the last 3 or 4 times, we really hope that we could have this event in person. Just before the event, the speakers and the organizers, we were discussing about the free pizza and how much we miss it. But unfortunately, we cannot give you pizzas today. But we have two excellent sessions by two speakers which we're really looking forward to. So let's start the event, and I will quickly share my screen. So I hope you all can see my screen. So welcome. Today is the 18th event and the fourth online event that we are hosting by PyBerlin. We are almost 2,200 plus members now, and we're really excited. Our community is really growing, and thank you to each one of you for being part of this journey. So why PyBerlin? Our main concept is that all of us love Python. We code day in and day out in Python, and we love to share whatever we've learned with you all. We're all based in Berlin, and we're welcome to everyone who is interested. Our organizers are Matteo, Anastasia, Miguel, and myself, Shreya. And thanks to everyone for joining. This is our third, or I think it's our fourth online event. And we do not have many rules and regulations, but one thing we want and expect from all our speakers and our audience and our organizers is to be nice, to be kind to one another, be patient, and respect the other person who is presenting or asking or attending the event. And there are more rules and regulations. Some of are available as well. And if you're interested, you can see the link on the page and read our code of conduct. It is very safe to ask questions. There, feel free to ask any big or small question you have. No inhibitions, no feigning surprises, no well ifs, actuallys, and just ask a question whenever you have one. You can use our links as well, which I will be talking about soon. So today's agenda, what do we have for today? We have two speakers. The first one is Dr. Karsten Yan, and he will be speaking on epigenetics. And our second speaker is Fabian, and he would be talking about how to build an interactive dashboard with Bokeh. For the questions, like I was talking about, you can use Slido and you can use the links mentioned on the screen, and the event code is also mentioned here. All these details are also present on our meetup page. So just in case you lose these links once we are in the event, feel free to use the meetup page. Or you can even ask your questions on the Twitch chat section, and we will ask them to our speakers once their talk is over. We're actively looking for speakers. So if you're interested to present your ideas, project, any personal project as well, then please feel free to contact us. And by speakers, we don't just mean professional speakers, but we are looking for people that have something to share. Even if you're talking for the first time, or if you just want to practice your presentation skills, then feel free to use PyBerlin as a platform to present your ideas. We have different formats. You can have a proper talk for 30 minutes and have a question answer session as well if you want. We also have small lightning talks of 5 to 10 minutes, and then a coding slam that is of maximum 5 minutes. In all of this, if you are comfortable to ask questions, for sure we'll make it possible for you. But if you're not comfortable and you just want to, like I said, practice your presentation skills, then we also have the option to not ask questions. Coding slams are just Python snippets or Python libraries that you can share with the audience. If you are interested, we're also super happy to provide mentorship and feedback, so feel free to reach out to us. And also, right now, we are looking for organizers. So a couple of our organizers have decided to pursue different options, and we are looking for organizers. We're really thankful to both of our organizers who would be moving on maybe, but we're always looking for organizers, and if you're interested, then feel free to reach out to us. If you want to submit a talk or proposal, this is the QR scan. This is the QR code, and please scan it, and the link is also available on your screen. And thank you for being part of this session. We will be tweeting throughout the event on Twitter. Our handle is PyBerlin Python. If you also want to tweet, please do so. Talk about what you found interesting or whatever you want, and don't forget to tag us when you tweet about our event. And also, this beautiful presentation and the logos and some other details, they were created by the following people as mentioned on the screen. So thank you to all of you who helped us create our logo and stories. And with that, we welcome our first speaker, Carsten Yan, who will be speaking on epigenetics. Carsten, over to you.
Thank you, Shreya, for the nice introduction. I will just share my screen now. So I hope it works. Does it work? I think so, yeah. So, yes, so my presentation is titled Epigenetics, as was already said. This is my capstone project for my data science bootcamp, which I did just at the beginning of this year as the pandemic hit. And you can see here the GitHub link to my repository where you can find the notebook I will present later. And you can also find the link on the PyBerlin meetup page if you want to follow along during the notebook section. And yes, so let's just get into the table of contents. I will talk about some theory first, because since I'm a molecular biologist by trade, and my project deals with data from molecular biology, I have to talk a bit about molecular biology instead of Python at first. Then we will go to the notebook, and then I will close with some short future works and closing remark sections. So let's delve into the theory first. These are the 4 bases of the DNA. They are called, let me just get the pointer out, adenosine, cytidine, guanosine, and thymidine, or A, C, G, and T for short. Almost every genome of every living being on Earth is coded in these 4 bases, and the specific sequence of these 4 bases code for the specific proteins that make up these living beings. So this is, as one could say, or this are the basic stepstones for the blueprint of almost every living being on Earth. But that's not all what these bases can look like. These bases can be epigenetically modified. Epigenetic modifications deal with the, as it says in the name, with modification of these DNA bases. On this slide, it's the takeaway you should have is not what these modifications are called or how these look like. You can see these here in light blue. There are different kinds of modifications. But what's important here is that although these bases are modified, these are all still read as cytidines from enzymes in the human body or in bodies of other animals, as is the case. These still code for the same things they would normally, what the C would normally code for, but these are still modified. And what this can do is it can change the accessibility of the DNA. It can change protein expression patterns. So what you really should take away here is DNA can be modified without losing its identity, but with gaining some other properties. So why am I talking about this, and why is this important? So as I said before, epigenetic modifications are relevant changes to the genome that do not alter the base sequence itself. But these modifications are, amongst other things, involved in the development of cancer and in evolutionary biology. So it's really important for researchers in labs to be able to recognize these changes and to efficiently research them. But currently established methods of detection are comparatively slow and complex. So what I did in this capstone project is that I tried to build a classifier to be able to classify these or to be able to predict these epigenetic modifications from a new source or from a newly developed technique in DNA sequencing. and I will talk about this technique in the next slide. This technique is called nanopore sequencing. What's important here is that there's here in gray, you can see here a so-called impenetrable membrane. During this membrane is closed tight, and DNA strands cannot pass through this membrane by itself. Inside this membrane, there's a very small pore. It's like a small tunnel. It's just big enough for a single-stranded DNA strand to pass through. And at the same time, there's an electrical current applied to the membrane, and this current can be measured, as seen here. If the DNA passes through the small tunnel, this electric current changes. And these changes in current are specific for each base. You can see here a G, T, C, A, and so on. What you can use this for normally is to identify these bases, to sequence the DNA, and to get the specific sequence of the DNA strand passed through. What I hope to see in my project, and what we see later if it worked or not, is if the epigenetic modifications I talked about before also change the DNA, the electric current measured, so that we can use the measurements from this nanopore sequencing technique to build a machine learning based algorithm to be able to predict these modifications in new and not yet researched DNA strands. So, and the databases for my project or the data set contains multiple reads, so multiple measurements of 5 different DNA strands of a length of 200 bases or around 200 bases, which were either synthetically modified or left unmodified. So I have multiple measurements of 5 different DNA strands, and these measurements are from strands that were either modified exactly in the same position each time for each of the different strands, or left unmodified. So that's all the molecular biology I want to talk to you about today. I hope I have not lost you yet. So next we will go through the notebook. That's more Python code, or that's at least some Python code. So here you can see the notebook. I will zoom in a bit. So first, here's a link again for the GitHub repository. Again, if you want to follow along, you can go here to see the notebook for yourself. At first, we have to do some setup, import some packages. These are the first block here is just for data handling and plotting. Second block, I defined some parameters for the plotting again, some stuff like line width and color of the plots. Let's just fold it in again. And the second block, the third block is one of the most important blocks of setup. It's importing some modeling packages from sklearn, which we will use later to apply the machine learning techniques. With that out of the way, we will load the data. Here with the standard pandas read CSV function, and take a look at the data, what the data looks like. The data looks like this. So we start with the column modified status. This is the target value. So it's a 1.0 if the specific base looked at here at index 0 in this case is modified. You can see here base 0 is marked as modified, and it's 0.0 if it's not modified. Then we have an identifier for which of the 5 DNA strands we're looking at. They are numbered from 0 to 4, so it's ref 0001 0 to ref 0004. And then we have the numerical data gained from the nanopore sequencing. These are a few columns, and there are a few different measurement techniques, and what these features mean by itself, I will not go into detail yet, as it's not really that important for modeling later. Just know that there are several features we will use to try to predict if the base is modified or not. So at first, we looked at the data, but now we want to have a deeper look at the data. How is the data set? Or what are the attributes of the data set we are looking at? So we will do a small exploratory data analysis. Therefore, at first, we will look at the target value distribution. This is important to look at because you want to know if your data set is balanced or imbalanced. And this is important to know later for handling the data, for choosing target metrics, and for choosing algorithms. And as you can see here, over 95% of my data is marked as unmodified, while smaller than 5% data of my data is marked as modified. And this is important to keep in mind because if you can imagine if my machine learning algorithm learns from mostly unmodified examples, it might have a problem to predict the small number of modified samples. If that is a problem or not, we will see later. But this is important to keep in mind. The next thing I want to show you is an interactive plot for further looking at the measurement data of our data set. Therefore, I import the interactive functions from Ipywidgets and prepare a smaller data frame used in plotting functions. Next, I define 3 helper functions for plotting. These are rather big matplotlib functions where for time's sake, I won't go into too much detail here. If you want to get a closer look at the code, you can go in the notebook yourself if you want. But where I want to draw attention to is how to build this interactive plot. So interactive plots with Ipywidgets are composed of so-called widgets, and you have to combine you can combine a few of these widgets to be able to interact with your plot directly. For that purpose, I prepared the play widget, which allows me later to play my data in a loop. And there you have to define some parameters like starting and end values, and what these values do, you will see in a few seconds. The interval you have to wait between each iteration, and what you want your description to be. Then I defined a text widget, an integer text widget, to be able to directly manipulate the place of data I'm looking at, and a toggle widget where I can switch between the different DNA strands, which were labeled from 0 to 4, as I said before. Then you can combine different widgets into one widget, where you will, in this case, I combined the play widget I defined before and the text widget into one box. And then I linked these 2 widgets, which means is if you change one, you will also change the other. Then you have to define a graphical output, which is done here with the plotting function shown before. And there you pass the arguments which are controlled by which widget. So the I argument is controlled by the play widget. The ref argument is controlled by the toggle widget. And then you have to display, or you have to call the display function to display all these. And what this does is the following here. This is a closer look at the data frame, or at the data we are looking at. And what you see here is here's the play widget where you can click on play to loop through the data. Here's the integer text widget where you can directly manipulate where you want to look at. And here are the toggle buttons where you're able to switch between the different DNA strands. And what you see here in the plotting is that you have on the left the modified version of the current DNA strand you're looking at, and the unmodified version of the current DNA strand you're looking at. And then you can see different measurements done with nanopore sequencing and the current sequence at the bottom. And what I want to draw your attention here is the top schematic representation, where you can see the DNA strand. And you can see what the nanopore actually reads. The nanopore, the pore where the DNA passes through, actually has a so-called reading frame. Those are the 5 bases that are at the tightest part of the pore and contribute to the measurement seen below. But what's important to know in this data set is that the base is only marked as modified if it's currently, or if it was measured in the center of the nanopore, marked here in blue. And what you also see here already is the red line here. Red lines indicate in this picture when the base is modified. So as I click on play here, the data loops through one by one, and the sequence you can see at the bottom shifts from right to left. This is, so to speak, a graphical representation of what the nanopore actually sees. And what you see here is that a modified base is approaching, And let's just stop here. No, one further, please. Stop here. And what you can see here is the bars here represent, as I said, different measurements taken by nanopore sequencing. But these bars represent the measurement as if the base was in the middle. So you can see here, if this base was in the middle, the modified one, the data would be this high. But what I want to show you also, that's why I drew the reading frame here, is if there's a modified base in the reading frame, you can see that the levels or the measurements are already rising. So the modified base has an influence on the measurements of the bases 2 before and 2 after it. This was also very important to keep in mind for data modeling later. And I can continue this graph just to take a smaller, or just take a further look at it, and we can see here, as I said, what the nanopore sequencing machine actually sees, so to speak, as you pull the DNA strand through the pore. And you can see here it takes some time because it has to render each plot each time there's a new iteration. But if we wait for hopefully just a few seconds, there it is, another modified base is approaching. And you can appreciate here again that the measurement data rises again a bit and falls again a bit. But what's important here is if we wait, if it's tilted in the center, right now, you can see here that the difference between the modified and unmodified strand is biggest when the modified base is in the middle of the reading frame. And that's what we hope to see, and that's what will hopefully allow us to later be able to build a classifier to predict modified bases. So now we have taken a look at the data. Now we want to build the classifier, and building a classifier means choosing an algorithm. And choosing an algorithm is not that easy because there are many different algorithms you can choose from, and you don't know beforehand which algorithm fits best to your data. So you have to compare different algorithms, and to be able to compare these algorithms, you have to have a numerical representation how well your chosen algorithm does on the data set you have before you. And these measurements are so-called metrics. These metrics can take many different forms, and I will just talk about a few of them and go into detail on one of them we will use. This is a schematic representation of a so-called confusion matrix. It's often used in classification problems as this one, where you have to choose between true or false, modified or not. And in this case, confusion matrix can only be done if you have pre-labeled data. So if you have in your test data, you know if it's modified or not. And you can see here the actual modification in true or false, and the predicted modification of my chosen algorithm in true or false. And you can count each label in a data set, comparing the actual modification and the predicted modification, and put them in the confusion matrix. And if both are true, you have a true positive. If you have an actual modification, but your algorithm says it's not, you have a false negative. You have a false positive if you have no modification, but your algorithm says it's modified. And you have a true negative if you have no modification and your algorithm already says it's not modified. And you want, as you might imagine, you want to have as many true positives and true negatives as you can, but that will not always be the case. From these counts, you can infer different metrics. And what we will take a look at today is the F1 score. The F1 score is comprised by the recall, which is the true number of true positives divided by true positives plus false negatives, which essentially says how many of my actual positives can my algorithm predict as positive or as modified. And the precision is the true positives divided by true positives and false positives, which means how many of my positive predictions are actually positives. And the F1 score, which we will use later, is the combination of these two metrics, which aims at balancing between precision and recall. So with that out of the way, we will get to the actual modeling. For modeling, you have to split your data set into a training data set and a validation data set, which I did here according to the DNA strands. Then I defined a random seed, which is used for modeling. And then I defined a random variable, which is used for training. And then I defined a random variable, which is used for validation. And then I defined a random seed, which is used for modeling. Then I defined a random variable, which is used for training. And then I defined a random variable, which is used for modeling. prepare feature lists, which are all numeric features in the data set to use later in the modeling process. And then I split these data frames again into an X and a Y frame, X containing the numeric data, Y containing the labels, the 1 or 0, if it's modified or not. And then at last, I defined a cross-validation strategy, where cross-validation is a process where you test a model multiple times, average the scores to see if your model can generalize over data or not. So the cross-validation strategy is defined. Next, get into the modeling itself. At first, I represent a small baseline model, and a baseline model is always important to do to compare your actual algorithm against. Baseline model in this case is just predict everything as unmodified. Then I plug these predictions into the confusion matrix I showed you as a schematic before, which can be done with the scikit-plot functionality. This is done here, and you can see here that we have confusion matrix with a high number of true negatives and a small number of false negatives. And in this case, at the first glance, this would look like a very nice predictor, because we have a high number of correctly predicted samples and a low number of incorrectly predicted samples. But in reality, this would not be very useful, because we want to see, well, we want to know, want to predict the modifications, where they are, and to see where they are. And this would not help us, because it would always predict as unmodified. And this is represented by the F1 score, which you can calculate just like this. And you can see that the F1 score is 0.0. So 0 means it's not useful for predicting anything here in this case. So we will have to use real machine learning algorithms. And for that, I built a small model selector class, which can be seen here. What I want to draw attention to you in this class is in the initialization function, I pre-programmed the scores, and I pre-programmed the algorithms to use. What these algorithms in detail are, if you want to know more about that, I can tell you later during the questions part. But what I want to show you is that it's very modular. So you can, in this case, I have only activated the F1 score and left in the logistic regression, the XGBoost classifier, and the Gaussian naive Bayes classifier. And but you can add or remove models in this class if you want. Just matters how much or how complex you want your project to be. So then the model selector class has 3 different functions. At first, it's a select model function, where each model is cross-validated on the data set according to each metric you chose. Then you can plot the selection, where you plot for each here in a for loop, you plot for each test or for each scoring you did, you plot the box plot to compare the different algorithms you chose, and a get scores function, where it spits out a Pandas data frame with all the scores for further analysis. So without further ado, let's define this class, and let's see it in action. Here I instantiated my class, named it combined data frame, inputted my cross-validation strategy, and then I select the model. This will take a few seconds, because it has to cross-validate each model on each data set. But this is done now, and now you can see the plot selection function. And this looks like this in this case. So here we have the combined F1 scores of our cross-validations for the logistic regression, the XGBoost, and the Gaussian naive Bayes classifier. And this helps you choose the algorithm you want to use for your data. So in this case, you see the logistic regression does very poorly, only about a F1 score of 0.15. The Gaussian naive Bayes classifier is a bit better, around 0.7. And the clear winner here is the XGBoost algorithm, which almost achieves a perfect score of 1. But if you say now, maybe let's see another metric, and maybe let's add another algorithm to the mix. We can just uncomment, let's say, the recall here, which I also talked before, and just chose uncomment another classifier, instantiate the model selector again, select the model, and plot the selection again. What you see here now is that we have two box plots, one for recall, one for F1 score, and you can compare the models against each other again. And you can see here, if you look at the F1 score, the random forest is pretty much this has pretty much the same scores as the XGBoost model. So in this case, you could say, maybe I would choose the random forest because it's easier to interpret, or it's nicer to use, or you chose the XGBoost algorithm. That's up to you in this case. Then we'll just use the getScores function at latest, and you can see here, if we look at the F1 score, XGBoost and random forest are almost the same. Here, there happened a funny thing with the Gaussian naive Bayes classifier. It has a perfect score of 1, and if you see something like that, you should always get suspicious that maybe it did something wrong, or it couldn't handle the data in the correct way. That's kind of weird in this case. But it happens from time to time. So at last, let's go to the evaluation of our classifier. In this case, I chose the XGBoost classifier. I instantiate it. I fit it to my test data and do a prediction on my validation data. And then I input everything in the confusion matrix I did before. And as you can see here, we have correctly predicted all 26 of our modifications as modified and only have 2 false positives. So in this case, we built a pretty good classifier for classifying epigenetic modifications in our data set. And with that, you can see that it's possible, or the summa summarum of my project was, it's possible, at least from this data set, to build a classifier to be able to predict epigenetic modifications. So, and with that, I am finished with my talk about data science modeling on epigenetic data, and I will return to my presentation. So this was the notebook. Some small future work sections. I thought about deploying neural networks on this problem to further improve the modeling accuracy, or the F1 score in this case. Another idea would be to implement unsupervised learning to be able to analyze unlabeled data sets, because as I said before, in the approach I used here, I have to have a labeled data set to be able to compare these algorithms to one another. Then what would always be nice to have are additional data sets to help my machine learning model to generalize better. Then what would also be possible is to port everything into cloud services to improve modeling times. And the last thing here would be the deployment, where I package my prediction models for application in a real workflow. So with that, I will come to my closing remarks. I want to thank Neue Fische and especially Larissa and Dirk, who were our mentors in the bootcamp, for that great bootcamp that allowed me to learn all the data science skills demonstrated in this capstone project. I want also to give a big thank you to my data science cohort, who made it a pleasure to endure this bootcamp with. And here you find, if you want to get in contact with me, you can find me here on LinkedIn. And here is also the GitHub page for the whole capstone project, if you want to have a deeper look into this data set. So with that, thank you for your attention. And are there any questions?
First of all, a huge round of applause. And thank you so much for the session. And Miguel, do we have any questions?
No, we don't have any question from the crowd right now.
All right, so we'll take a 10-minute break. Even if you all have any questions, meanwhile, feel free to post it on Slido or on the Twitch chat channels. And let's have a 10-minute break, and we'll see you back in 10 minutes, say around at 7:55 p.m. So, hello everyone, welcome back again, and very happy to have you. And let's welcome our second speaker, Fabian, and he will be talking about how to build an interactive dashboard using Bokeh. So, welcome, Fabian, and over to you.
Thank you, Freya. So I will just share my screen and start the presentation. So here we go. So thanks for having me, and thank you all for your time. I'm Fabian, and I'm here to tell you how to build an interactive dashboard with Bouquet. So as mentioned, my name is Fabian Workmeister, and I'll do this in collaboration with PyBerlin. So what are we going to look at today? So the topics are all around Bouquet. So at first, I will tell you exactly what is Bouquet. Then we go further into some basic code, and I show you some examples for static plots, all within a Jupyter notebook. Then we'll go further into detail into more advanced code, and I'll also show you some examples for real interactive plots. And to round it all up, I show you one fully interactive dashboard I've built for my final project. And I also give you a hint how you can web host your own project on a website. And finally, I'll tell you where you can learn Bouquet, where you find resources, docs, and materials, and so on. So let's jump right into it. At first, let me introduce myself. As told, I'm Fabian, I'm 29 years old, and I would describe myself as an autodidact and a data enthusiast. Autodidact because I self-taught me Bouquet, and now you can all profit from it. So a little bit to my background, I graduated from university with a master's degree in business administration. Then I worked for 1.5 years in online marketing and market research, but noticed that I wanted to do the dive deep into data. And that's why, exactly as Carsten did, I graduated from a bootcamp called Neue Fische in Hamburg. I graduated in May, and I was in the same class as Carsten. So, and you can see my final project at the end, as Carsten also showed you his. And a little bit further details of me. Yeah, my personal interest is in football and gaming data. I look forward to also become a data scientist, and I'm eager to learn new things every day regarding data, data science, and so on. So let's start with the topic. What essentially is Bouquet? So if we take the self-description from the website, and it says that Bouquet is an interactive visualization library for modern web browsers. It provides elegant, concise construction of versatile graphics and affords high-performance interactivity over large streaming data sets. So you see the emphasis here on this interactivity. For example, if we look at other visualization libraries such as Matplotlib or Seaborn, you probably know this, then you know that you can build static plots, or as Carsten showed you, a somewhat interactive plot, but with Bouquet, there are a lot more opportunities. Another visualization library, which is pretty common, is Plotly. I didn't do the deep dive into this. This would be next step, but as you know, we're currently talking about Bouquet. So let's have a look at the basic code structure of Bouquet. So as you might know, everyone, we start with importing the libraries from Bouquet, and so we import. Then in a second step, we kind of create an empty canvas of which we draw. So we say our plot is called plot, and we call a function called figure and have several arguments within it. And in the second step, we actually draw on our canvas we created here. So we say we draw on this canvas, and we draw for example, we draw some circles with several other arguments. Then in the third step, we can customize our plot. Like, for example, if you see here, where is the location of the legend, or what text font we want to have for our label on x-axis. And in the final step, we show our plot in our desired layout. So we can build a layout which is also a function here, and then we show the whole layout. So we arrange different aspects of the plot. So we start with the canvas here, and then, like Bob Ross did, we paint on our canvas. And as I did in my progress with experimenting with Bouquet, I didn't make mistakes. I just made happy little accidents so that I could learn further. So I have some basic examples for you with simple data, with the iris data set and the Wirecard stock value. I show you the output, but we'll have a look at it right in the Jupyter notebook. So to get a hint how Bouquet actually looks like, this is the iris data set, and this is the Wirecard stock value, as you all might have heard in the news. There were some big troubles here. That's why the stock value dropped significantly. Another example would be to have different layout options. I told you that you can create your different layouts, so we can arrange plots within our layout. And we also can have some kind of tabloid view, but just that you get an idea, I will show you it right in the notebook afterwards. And we can also customize our plots. We can add colors. We have some different plot tools like you see here, and we can incorporate legends. So, like I told you, we'll jump right into the notebook. So basically our contents here are that we look at the libraries, create a canvas, show some basic examples, show the layout options and the advanced customized options. I will show you all which I just showed you. So we start with our libraries, as you know, Pandas, NumPy, and some Bouquet libraries. By default, Bouquet output is shown as an HTML output right in your browser. But in our case, I want the output should be shown in my Jupyter notebook. So we call this function here. And if we would reset to HTML output, we could simply uncomment this one here. So we start with creating a canvas. So basic is we call our plot empty1 for empty canvas, call the figure function, and call the show function. And we see here, this is our empty canvas. Now nothing is drawn on it. We also get the warning here, there is no renderers, there is actually nothing on the plot. What can we customize about this? So we create a second plot. We can give the whole thing a title. We can label x-axis and the y-axis. And we can also define some width and height for our canvas. So same as before. So notice that now we gave some arguments here about the size. The size was resized, and we got a title and some labels for our axis. So far so good for our empty canvas. Now let's draw something on it. So let's start with some random data and start with creating a canvas on which we draw. So it's basic as this code here, so nothing new to you. And then we can manually generate, for example, some data like some x values and some y values. You can use integers and floats, no problem. And then we draw on our canvas. So we have to define, we want to draw on plot 1, which we defined here, and we want to draw some circles, and our x values should be our x values, and y should be our y values. And then we simply call show our plot, and you see, simple as that. Now we plotted these data on our empty canvas. So as I noted here, you are not limited to using circles. There are several other options available, such as crosses, or you can use squares. For example, if we try squares, you got some squares here. So simple as that. If you want to know what kind of drawings are available, you can have a look at the documentation here. So you might all know this famous iris flower dataset. And with this, we can show you that Bouquet can easily handle Pandas data frames. So Bouquet already incorporates the flower iris dataset. So we download it right here. And then we build another plot, same source code as above. We plot some circles on it. And you see we can use Pandas notation. So we use our iris data frame and say we want to have sample length and sample width as x and y. As you notice here, you don't have to specifically say this is x, this is y. This is the if you plot it in this this sort of thing, it automatically assumes this is x. and this is Y. So let's see, we got our iris data set here and everything works pretty fine. Some small adjustments we can make here. For example, we want to have our circles at green and we want to have a bigger size. Simply do this and you see simple as that. Third example, we use Wirecard DAX stock data, which I sourced from Yahoo. And I downloaded here. We only use the date columns and the closing value columns. And we have to convert the date column to a data and variable format so that Bouquet can work with it. So you know this already. What we can do now here is that we can draw multiple items on one canvas. So as you see here, we draw some simple lines between each data point. Let me just out-comment this. So we start, so we have some lines here. And to make it look even better, we can additionally plot some circles on it. So to every data point. And now you can see, you got these, each data point is more clear to view. And yeah, so you can simply use these data. You can use data formats. So no problem. If we come to layout options, Bouquet layouts, they can be seen as a grid visualization. So it consists of rows and columns. And these are also the functions called. And let's say we want to plot 1 and 2, which is basically this plot and this plot. We want to have both plots in rows or in a row next to each other. So we simply say layout 1 is row consisting of plot 1 and plot 2. And then we show layout 1. And you see these are both plots next to each other. If we want to have them in the column, it's basically the same. Instead of row, we call column here, and you see we got one column with plot 1 and plot 2. You can combine this. For example, we want to have our plot 3 with Wirecard below a row of plot 1 and 2, like in our first layout. Below would be this. And you see we got plot 1 and plot 2 and plot 3 here. So you see you can combine existing layouts, which we defined here, into a new layout. So let's come to panel views. This is also pretty easy. You can switch between different views all within one layout. So you import these functions here. You define the 3 panels which you want to show. So simply as that, you call the function and you say, in our panel should be shown plot number 1, give it a title, and then incorporate all into one layout. Pretty easy. And what you get with is here. So now we got all 3 plots and can switch between in a tabloid view. So pretty neat. If you have many plots, this is really cool. And coming to advanced customization options, there are a lot. Trust me, and in this section, I just want to give you some inspiration and an idea of what is possible. So we use data from the World Happiness Index to illustrate some of these options. You can find the data set here. We import it with Pandas and have a look. So basically, these are mainly all countries from the world, and they get a score based on gross domestic income, based on social grades, health expectancy, freedom, corruption, and such, and you get a basic score. So in order to process data for advanced customization in Bouquet, you can create a so-called column data source. So to explain this, you simply call the function and then use your columns from your data frame. For example, we got happiness data frame and overall rank. So this is this column here, and you simply assign it a new name here. And from now on, we can refer to these self-defined names on the left, so rank, country, and so on. And be aware that when you plot now with this column data source, you always have to incorporate the source object, so this one here. But it's pretty easy, and it's necessary for advanced customization in Bouquet. So let's try this. So we built another plot, and with basic source code as above, and now we plot some circles on it. As you might see, now we can refer to these self-defined names, score and GDP, and give it some size. And now we refer to the data source, as source is equal to data source right here. And now we can customize our axis. For example, we say we want to have our tick formats as dots, and that we have like 1 decimal point, 2 decimal points. You can adjust some axis label, text font size. You can have some font actual Arial here. You can have some text color, some font style. You see there are so many options just that I can give you an idea. And if we plot all of this, you see now this is blue, this is red as indicated here. We have different fonts and so on. So try to experiment a bit with it, but it's pretty easy. What you can also incorporate, you might have seen it on the right here. This is a Bouquet toolbox. We also got many tools, which you can see here. I already show you some here. I define them beforehand, and then we simply pass them to our figure and say, tools, we want to have our toolbox here, and the toolbar should be on the right. And as above, we plot our plot, and now we can interact with our plot here. You can choose different options so that you get an idea. We can also incorporate some tooltips for selecting data. So same as above, same as above. Now we incorporate a hover tool. It's pretty easy. You can say, once I hover over some data points, some things are shown here. And we simply assign our column data source, which we defined here, and say, I want to have this right here. And simply incorporate it, and now we have our tooltip and can hover over different countries, for example, with respective data we defined here. Finally, we can also do some color mapping and add some legends so that you can distinguish between, for example, categorical data. And so to show you customized color mapping, same as above, build our figure here, and then we import our category color mapper, and then we can use our continents, which are defined in our data frame, and we can assign some colors in this order. And same as above, instead of color, we now pass our color mapper as a dictionary. We incorporate a legend and plot the whole thing. And you see now we got all these colors referring to each continent of each country. So this is maybe a legend isn't placed very good. So what we can do now is customize our legend. So we can, same as above, incorporate a figure, color mapper, draw our circles. And now, as you've seen before, you can now customize your legend with location. You can adjust font style, size, and so on, just to get an idea. Now we place it here, bigger size, and so on. You see it. Pretty simple, just a few lines of code, and very neat. And to show you finally what legends are capable of, I show you some interactive legend where you can play around with the data. So I'm not getting into detail here how to build this, but you can have a look in my notebook afterwards. What we simply want to do is that we want to click on our legend, and then this data is hide. So you incorporate these two functions, pass it to our figure, and then we draw 5 independent data sources on it and tell our legend we want to hide on click. And so you see here, we click on Europe, and Europe data disappears. We click on America's data, and America's data disappears. So pretty neat, only a few lines of code, and you can do some impressive plots. So back to my presentation. So we ended here, and oh, sorry. So, and let's jump into more advanced code. So it's the same as above. We got our basic codes with figures and drawing on it. Then we built some interactive widgets where we can interact with our plot. And finally, we have to build a function which updates the plots based on the user input. And then we have to arrange widgets and plots, which we build here in the layout. And then we have to start the whole app via a console command, which I will show you right now. So basically, build your canvas, draw on it, then build a function for updating, and finally, run your application via your console command. So it sounds difficult, but it's really easy. So what I want to show you now is how to build an advanced interactive plot with a Pokemon dataset, which I'm pretty interesting about. So I got all these steps, got them here, but I think I show you the final product right here. So we got an interactive dashboard here. Our data source are stats of the first 2 Pokemon generations. And we also want to add some small images to each Pokemon as a tooltip. So we start right here. We import our libraries. We import our data source and push it as a column data source. So that's worked fine. So, and now to our basic code for a bouquet plot. In order to incorporate little images to our tooltips, we need to write a little HTML or CSS. So this was new for me, but Bouquet already got us covered with a short tutorial. So pretty neat. I only copied this source code here, and it worked really well. So don't be afraid of difficult tasks. Just look for a tutorial, and you get used to it. So we import our figure and hover tool. We say our hover tool should consist of a small image, our name, overall strength, and overall health points of our Pokemon. So then we define a toolbox. We draw our figure just like before, and then we plot circles with the attack and the defense power of each Pokemon. So let's give it a try. This isn't shown right here, so I'll show you in with my command console. And now let's look at the code for our interactive dashboard. So what we need to do is here, we have to build our widgets from previous examples. As you can find here, you find them on my GitHub if you want to learn it step by step. So for example, we want to have some sliders. We want to have some checkboxes where we can tick on or off several main types of Pokemon. We can have a text box where we can search for names or substrings. And we can also switch between generation 1, generation 2, generation 1 and 2. And as I told you, we built a function. In this case, we built 2 functions. One function selects a new data frame. So because when a user gives input to our plot, then we have to update our data frame so that the new data is shown. For example, we got some boundaries here, which we have to incorporate. So just have a look in it and give it some time. Can be pretty confusing at the start, but I think you get used to it with all my examples. So you select a new data frame based on the user input, and then you update your plot in a second function. So you basically say my new data frame is in this function. We get a new data frame with updated data, and then we pass new data to our plot, and all is updated. And then we have to actually pass our data to these functions. And finally, we add all these widgets. We add to our plot. So we want to have a row. So this means we want to have our widgets on the left side and our plot on the right side. And then we add everything together. So, and now to show you how this all works, I already said it here. You have to use your command line. Everyone has it on Windows, on Mac, on Linux. So there's no problem. So you have to type in Bouquet serve and then your IPython notebook or your .py data file. Oops, oh, sorry. To navigate to my desktop, and then we say Bouquet serve this one here, and then we say show. Then we let Bouquet work a bit, and now we have our interactive plot here. So we see here our tooltips work perfectly fine. For example, we got Onyx and Golem and so on. You all know them probably. I had a lot of fun creating this dashboard. And you can switch between generation 1 or 2 or both if you want to. Let's stick to generation 1. You can say my Pokemon should have at least, I don't know, 86 health. So this is pretty much everyone with health above this. I want to have an overall strength of this one, so you can simply adjust your data which is shown. You can select here the main types of each Pokemon. So for example, there are only a few left here, and you can also, if we take everything on again, we can also search for some substring. For example, we all know him, Pikachu. All right here. And so you can build your interactive plot very easily and very comfortably. Oops. So let's switch back to our presentation. So you have seen how you can build an interactive plot example. Let's talk a few minutes about my final project at Neue Fische, like as Carsten did. So my business case consisted of football player and their market values. So football players are an investment object in terms of market value, since they can be sold or bought by football clubs. And estimating and predicting these values is of crucial interest for football clubs in order to evaluate own and potential new players. Some of you might know Transfermarkt. This is a platform on the internet which tries to evaluate football players in terms of market value. They do this via crowd estimation. That means people can subscribe to this platform and discuss each other and discuss with each other if they find some estimations for football players. And you also probably know the video game FIFA 20, and I wanted to know to which degree are player attributes and ratings from this video game FIFA 22 able to perform these estimations and predictions. And so that you have a look at my project roadmap back then. So I started off with finding this business case. Then I did some data mining, probably getting data from FIFA 20 and getting data from Transfermarkt. And there was a huge problem because the player names weren't matching. I mean, you can probably imagine if you have like 20,000 players and they don't exactly match because they're written differently in the video game and on Transfermarkt. It was a real pain, and it took me like 1 week to actually solve this. So I took a pass back, did some data merge between both data sources, and then went on with some data analysis and some predictive modeling. And my final shot at my goal literally was to deliver this dashboard. So, and one of my main deliverables, as I told you, was a fully interactive dashboard. So you get a screenshot here, and it can also directly be accessed via this web page. So if we have a look at it, so you see I incorporated mostly everything I showed you right before. So it works with this legend here. You can have some data adjustments and so on. So works pretty well. And example, we got Thomas Müller here. And so, yeah, works everything fine. And yeah, you can have a look at it if you want to. So it's in my presentation. And yeah. So, and now you probably wonder, how did I get this on the web? It's also pretty easy. Might look difficult at first, but it's basically 3 easy steps. You create your own project and save your Bouquet code as a .py file. Then you choose a web hoster for your project. For me, Heroku worked really well and is completely free for small projects such as mine. And then you have to sync your local project file with Heroku via Git. This can be a bit tricky, but here I give you an article where there's a step-by-step instruction, which I personally found really helpful. So to conclude everything, where can I learn Bouquet? So yeah, the Bouquet website itself provides an extensive and comprehensible documentation. You got user guides with step-by-step instructions, and you also got several plotting examples where I got some inspiration from. Of course, you can look for courses on your preferred learning platform, Udemy, DataCamp, even YouTube got some. And you can also, what helped me a lot was to look for short online articles with Bouquet tutorials. I link you to articles which I particularly found very helpful and gave me a lot of inspiration. And obviously, you can learn such things in meetups like this one. So finally, thank you very much for your attention. This was basically me after I got everything running and loading, and it was a somewhat painful journey, but in the end, once you make this journey, every further task is pretty easy with Bouquet. So if you got any questions, feel free to reach out to me via email, LinkedIn, Xing, or my GitHub. And yeah.
Thank you, Fabian. That was an amazing and very interesting session. A huge round of applause. I really liked the case study that you showed with the Pokemon example. It's very interesting. And I worked with Bokeh before, and it was quite tiring, but the walkthrough that you gave did make it look at least simple. And I hope that those who are viewing it find it useful and find a use case in their projects as well. Miguel, do we have any questions?
So we don't have questions for this talk so far, but we have questions for the first talk. Maybe we can wait while we answer the question from the first talk, we can wait for the next question. I mean, question for the other talk. I don't know.
Sure. So Carsten, we have Carsten on the call, so feel free to ask your questions, Miguel.
So Carsten, a couple of questions for you. So the first one says, did you say you use some current measurements for detecting epigenetic modification to the DNA strand?
So I'm not sure what part of the talk it is referring to. So in the measurement, there's or in this DNA nanopore sequencing method, there's an electrical current applied to the membrane where the small tunnel is in there. And then you can measure this applied current and changes in this applied electrical current, if that's what the current is referring to. If current means new, then also yes, because DNA nanopore sequencing is a pretty new technique. I think it's like 2 or 3 years old at most. So it's a pretty new technique, let's say. But I didn't do this measurement myself. I got the data from the internet. So that's not, I didn't stand in the lab and measured these measurements myself and then applied machine learning to it. Unfortunately, that would be very nice, but yeah.
All right, I have another question here. And it says, in general, how do you choose what a nice measurement, i.e. what to measure is for a biological molecular process for modeling?
I mean, I don't personally have a real life experience in that I had to decide, okay, what do we measure for modeling? I mean, what would I say to that? What do I guess? Not guess, but what would I do? I mean, it's as always, or maybe as the reality is in the lab, you measure what you have. So you don't have every option of measurement available. And I would say for modeling, it's very important to have many different features to be able to, because beforehand, most of the time you don't know which feature is the most important one, what distinguishes most what I'm looking for in this case. We didn't know beforehand, okay, what does the or what is the most deciding factor that the modification influences. So I would probably say it would be a good idea to have measurements that measure many different types of aspects from your sample.
All right. We don't have any other questions, and also we don't have we don't have questions for the first talk either. So yeah, back to you, Shreya.
All right, I think we are reaching towards the end of the session. And thank you to both our speakers, Karsten and Fabian, for your time and sharing your work with our audience. It was really interesting. And thank you to all those who tuned in. I will be sharing my screen. So once again, thank you. Thank you for coming. And thank you to our speakers, Karsten and Fabian. It was very interesting to learn about genetics and to how to use Bouquet. The next meetup is on August 17th, and we already have 2 speakers lined up for the event. If you are interested, feel free to check out our meetup page. The details are mentioned over there. We're also looking for organizers, like we mentioned, and we would be happy to have any one of you who's interested to work behind the scenes in organizing these platforms. There's a lot of interesting activities like streaming or doing this kind of intro and closing sessions or asking questions, and we would be happy if you're interested to join us on this journey. We're also looking for speakers. If you're interested to give any kind of talk, a 30-minute talk or a 5-minute talk, feel free to reach out to us. The link in the QR code is mentioned on the screen. Even afterwards, you can find these details on our meetup page. We would love to hear from you. How was this event, or how you find our platform in general? So please feel free to tweet on Twitter. Our handle is PyBerlin Python. Don't forget to tag us. And also, if you would be interested to join our Slack community, the link is mentioned on the screen. It is pythonberlinslack.herokuapp.com. And once again, thanks for coming. It was really wonderful to have this session, even though it was virtual. And we hope that in the coming months, we can have one-to-one or personal live events as well and share some pizza with you. Thank you so much for your time, and see you next time.